Now we're going to be talking about here with the citric acid cycle. Keep in mind, this is going to be mitochondrial only. You know, the enzymes, coenzymes, the proteins, all of this is going to be centered in the mitochondria. Hell, most of the enzymes and the proteins we're going to be talking about here are translated from the mitochondrial's own genome. Okay, so this isn't going to be nuclear derived information for these. No, this is going to be in the mitochondria. So what we're going to be ultimately looking at is what it takes to get some electrons. Excited electrons, which are going to be, that energy is going to be bled off, pump some hydrogen so you can get some ATP synthesis. Now, a lot of what we talked about is going to be pyruvate entering in, going pyruvate, acetyl-CoA, but got to remember the oxidation of fatty acids. Go through, cut the fatty acids, cut the amino acids every 2 carbons. Well, that can be acetyl-CoA also. And we'll be discussing in this section and later in the chapter how that gets done. The complete oxidation, meaning we're going to strip off the electrons and we're going to consume the acetyl group. You know, releasing carbon dioxide. We're going not only after the excited electrons, but we're going after the carbon-carbon bond energy. Okay, maximum potential. By doing it in so many steps, we're also minimizing loss. So if we went directly after the pyruvate, or we went directly after the acetyl-CoA, there's just going to be too much loss. That whole second law of thermodynamics. Okay, for every time, you know, just that first law, you can't, what is it, energy cannot be created or destroyed, it can only be transformed. You can only take it from one form, transfer it to another. And second law says whenever you do those transformations, you're going to lose some, okay, to entropy. Well, if we went and took all the energy that's there in the acetyl-CoA and took it all at once in one big step, or, you know, a couple of big steps, we're going to lose too much, too fast. And you'll no longer be 37 degrees Celsius, 98.6 degrees Fahrenheit. You're going to be a lot hotter. So, a lot of what we're going to be doing here is making small steps so we can have energy conservation. Okay? Want to minimize loss. So we're going to be looking at moving the functional groups, putting them in position to facilitate oxidation or oxidative decarboxylation. So yeah, some of those steps are just basically going to be moving a functional group from carbon 2 to carbon 3. By doing so, it sets up so that they can go get, you know, an excited electron off of carbon 1, or you can go get the excited electron off of the carbon 3 now. So when you go through, take the acetyl-CoA, add it into the citric acid cycle, the first thing that's going to happen is you're going to form citrate. Citrate, again, going to be, you know, consumed, not all at once. It's going to be broken down, and you're going to yield some carbon dioxide, some excited electrons, and then a couple of triphosphates, GTP or ATP, and we'll talk why the or right there later. So first thing off the bat, citrate synthase is going to take the incoming acetyl-CoA. Here's our carbons from the acetyl group, carbon 1, 2. And here's oxaloacetate, 1, 2, 3, 4 carbons. Citrate synthase is going to do a hydrolysis reaction, breaking this bond, adding a hydrogen back, adding an oxygen or a hydroxyl technically right there to the carbon 1, And now you have 1, 2, 3, 4, 5, 6 carbon citrate. The delta G here, negative 32.2, this is an extremely exergonic reaction. We need this to be this low because the concentration of oxaloacetate is actually kept relatively low in the mitochondria. So we need to, whenever there's a free oxaloacetate, we need this to occur. We need that to occur now. If this was a high number, close to 0 or positive, well then the free energy, getting enough free energy, getting ATP or whatever to get this to push through, that would be the limiting factor. But by having it so low here, negative 32, it's the concentration of oxaloacetate is our limiting factor for this enzymatic reaction. What's interesting is because the oxaloacetate is the limiting factor, this enzyme has evolved to the point that it's the oxaloacetate that's not only acting as a substrate, but it's also acting as an allosteric activator. Normal conditions, citrate synthase, it has the shape that's near correct, but its active sites do not recognize acetyl-CoA until oxaloacetate enters into its pocket as part of the active site. Well, then it alters the shape of the remaining of the active site, that whole induced fit thing, and it now becomes the correct shape for acetyl-CoA to enter in. No oxaloacetate available, there's really no reason to take acetyl-CoA into it and do the cut and all that stuff for removing the 2 carbons off of the coenzyme A, because there's no place to put it. So, timing, concentration and timing are the thing here. Low levels of available oxaloacetate, well, that means that the citric acid cycle has been slowed down for some reason. It's a regulatory point. If the citric acid is slowed down, the citric cycle has been slowed down, well, maybe that means there's no available NAD, which means that there's no final oxygen electron acceptor in, the oxidative phosphorylation portion at the end of the electron transport chain. So the NAD's all stuck as NADH, and those few steps on the citric acid cycle where you should have that redox reaction occurring, well, there's no place to put, so they're stuck, which means there's no regeneration of oxaloacetate, and there's no reason to go through and keep pushing stuff into the cycle. So it's a great little regulatory mechanism. What we see happens in here is multiple amino acid residues in the active site. Oxaloacetate enters in first. It being there, all induced fit, changes the shape, allows for the acetyl-CoA to interact with histidine in the aspartate here. Nucleophilic attack here, rearranging of the bonds. Oxaloacetate interacts with histidine, what is that, histidine 320. Starts rearranging the bonds here on what is carbon 2 here. We get the nucleophilic and electrophilic interactions from the 2 carbons acetyl group here. Coenzyme A is pretty much stuck out here, formed an enol intermediate, but right now coenzyme A is not involved in anything. So, rearrange of the bonds here, the hydroxyl here, the hydrogen bonding with the histidine, that bond shifts over, so you get the double bond, the carbonyl group. This carbon-carbon double bond here shifts, and now this carbon 2 here is now going to interact with this carbon here, or this carbon 2. And the oxaloacetate is now bound here. Carbon 1, 2, 1, 2, and then 1, 2, 3, 4, 1, 2, 3, 4. And it's now hydrogen bonding here with histidine. So this histidine's now being ignored. We got this final carbon sulfur bond here, which water is going to be added in. Hydrogen's going to go to this sulfhydryl here. So that's now released, or to that sulfur there, so it's released as a thiol. The hydroxyl is going to bind to what is now carbon 1, giving us our carboxyl group. Sorry, the hydroxyl interacts with this carbon, what is now carbon 1, and you get a carboxyl group here for carbon 1. So here's our citrate. 1, 2, 3, 4. Carboxyl, carboxyl, carboxyl. Aconitase, or aconitate hydratase, is going to take our citrate and convert it to isocitrate. But it's not a direct conversion. It turns out there's going to be this intermediate known as cis-aconitate. Hydroxyl, hydrogen, hydrogen, hydroxyl. It's not just, hey, we're going to pick this one off, swap it over there, and then move that one over there. No, turns out it's actually more involved. Going to pluck off hydroxyl and hydrogen, 2 hydrogens and oxygen. Hey, there's water. Water gets removed. So now we get a carbon-carbon double bond here between these 2 carbons. This is our cis-aconitate intermediate. Aconitase removes water, and then it's going to grab a separate water molecule, add it back in, but this time carbon 3 gets the hydrogen, carbon 4 gets the hydroxyl. What's interesting is even though these are reversible reactions, it does seem to want to push towards isocitrate, because as isocitrate levels drop, we see less reversibility. So the citrate becomes more isocitrate when these levels start to drop. When you look at it inside the active site of aconitase, holy smokes, Batman, you get this iron sulfur box, this cube occurring here between 3 cysteine residues. This will interact with and twist and tweak our citrate molecule. Next step, we need to take the isocitrate and we need to turn it into alpha-ketoglutarate. This is Isocitrate Dehydrogenase. Dehydrogenase means we're going to be removing a hydrogen, so here's a redox reaction. But the thing is, we're also going to be doing a decarboxyl step, removing a carbon, a CO2 molecule. So we're going to be losing a carboxyl functional group. Keep in mind, these 2 carbons right here, these 2 carbons right there, are from the acetyl group that we are just tracking, that was just added 3 slides ago. So this is a pre-existing carbon that's going to be removed. So this was one of the carbons that was on the original oxaloacetate. So the isocitrate, NADP to NADPH. In the process, we see the shifting of the electron pairs. CO2 is cut, released. The carbon-carbon bond right there, now going to go carbon-carbon bond here. We see the this double carbonyl group is now going to interact with, what is it, sorry, misspoke, Mn, manganese. So we have an elemental manganese here that's going to act as in an intermediate structure here, so that we get this cyclic here, which is going to resolve as this carbon-carbon double bond is going to be shifted over here to become the carbonyl. This bond's up there to become a hydrogen. So we now have alpha ketoglutarate. Let me do this again. Here's the two that we've been tracking that just came in on the acetyl. Alpha ketoglutarate to succinyl-CoA. Remember, alpha ketoglutarate dehydrogenase complex, our E1, E2, E3 variants. Well, here's the substrate. Instead of pyruvate to acetyl-CoA, here we're taking alpha ketoglutarate and we're going to yield succinyl-CoA. Again, CoA, our coenzyme, and the dehydrogenase, we're going to take NAD and reduce it to NADH. In the process, we're releasing another carbon dioxide, another carboxyl functional group will be cleaved and released. So this whole mechanism, this whole process is a conserved thing, pyruvate dehydrogenase. We see it here, with the alpha ketoglutarate dehydrogenase, come up with acetyl-CoA, succinyl-CoA. We also can see it, for some other, where you take leucine, valine, and you turn them into CoA, methylbutyryl-CoA, which can then be fed into this whole process. Again, this is going to be, they think this is due to what is called divergent evolution. Duplication of genes or parts of a chromosome these genes have been on, were originally on in some common ancestor, accumulation of mutations at varying rates on the different copies. And what was the substrate for all of them? Pyruvate. Well, selection, evolution, now says, oh, it's alpha ketoglutarate, or it's some alpha keto acids, such as leucine, valine, or whatnot. So the succinyl-CoA, okay, got to change that up. Can't do anything with it because we can't stop here, and we can't directly turn this back into oxaloacetate. So first, we're going to remove the CoA. So we're going to use some substrate level phosphorylation, G-diphosphate inorganic phosphate, going to form GTP. Hydrolysis reaction, so we're going to cleave off the coenzyme A here. Going to break the carbon to sulfur bond here, add in hydrogen there, boom, going to keep going. I'm sorry, add in the hydroxyl there. So when we look at what's happening here, this is a pretty big process. We're starting with succinyl-CoA. First process in this reaction of the succinyl-CoA synthetase, going to remove the coenzyme A, and inorganic phosphate is going to be swapped here at the thiol group, the thiol group, so that we can release coenzyme A. This is going to be replaced by a phosphate group, which you can see in the slot in your handouts behind my big head here. Cleaving off that phosphate, going to be releasing the succinate. That inorganic phosphate, though, is going to remain bound to a histidine residue in the active site. Phosphatidyl, phosphohistatidyl enzyme, which is part of this succinyl-CoA synthetase complex, is then going to take that inorganic phosphate that's on this histidine residue, and it's going to transfer it to GDP. GDP now becomes GTP due to substrate level phosphorylation, and everything has been reset. You took succinyl-CoA, removed the coenzyme A by turning it into a phosphorylated succinyl intermediate. Cleave off that phosphate group by adding in a hydrogen. So you now have a free carboxyl, and you have our 4-carbon succinates now been released. That phosphate's now going to be added to a GDP through substrate level phosphorylation to yield GTP. You look at this figure here. This is showing, right here's the histidine that's going to be, phosphorylated. Here's the coenzyme A. Here's our 4-carbon strand here. You have these, what do they call them? Beta subunit power helices. Alpha beta subunit, beta subunit. These alpha helices, the way they're shaped. Remember we talked, what is that? Oh, not all of you are going to remember. Last semester when we were talking protein structures, we said that some of these secondary structures that form these alpha helices, they can, when they're formed, the way the R groups are situated and which R groups are there can give one end of the alpha helices a negative, the other a positive. So what you end up with is this polarity. This polarity is going to give an overall positive charge here for these two alpha helices, which is going to draw the negative charged 4-carbon carboxyl groups away from histidine, and it's going to aid in positioning and cleaving of the coenzyme A and all of that. Exactly how this works, I don't know. That's part of the biochemistry and the organic chemistry that's beyond me. I'm a cell biologist, microbiologist. If you really, really, really want to know, talk to Drs. Delgado or Dr. Hasty. They'll be able to help explain it better than I could. So what you end up with, remember, is that substrate level phosphorylation. You have what's called nucleoside diphosphate kinase. Okay? Nucleoside diphosphate kinase means A, DP, G, DP, T, DP, UDP, ADP, whatever. You start with those, it gets phosphorylated by what we just saw in the last. But the thing is, TTP, UTP, CTP, even the GTP to an extent, are not readily usable throughout the cell. There are a few enzymatic reactions that use the G-triphosphate version as, the energy for the reaction or the activation, but those are very few. The majority of all the reactions, as you've seen throughout this course, and if you took this fall semester course, you saw it was always ATP. So the nucleoside diphosphate kinase is going to take one of those nucleosides, that's a triphosphate, and it's going to transfer that terminal phosphate to a diphosphate and turn it ATP. This is a net zero game here, okay? It is both exergonic and endergonic at the same time, so they just say it's a push, okay? So it's a net result, 0. It's neither exergonic or endergonic totally. So the GTP, limited use, widely used. CTP, TTP, those, no use whatsoever. So now you've had succinate. Okay, what can we do with it? Well, Succinate Dehydrogenase is going to go, like the name says, it's going to remove a couple of excited electrons. This is going to get oxidized. FAD to FADH2. So FAD becomes reduced to FADH2, and those bonds that were the hydrogens are now going to reform, and you're going to get a central carbon-carbon double bond there. This is our friend fumarate. If you think back to the last slide, the succinate, 1, 2, 3, 4 carbons that had carboxyl groups at each end, 2 methyl groups, or 2 CH2s here in the center. The Succinate Dehydrogenase, which is going to remove one of this hydrogen and one of those hydrogens, it's a stupid enzyme, okay? It doesn't look at something and go, oh, yeah, I recognize that. No. What it does is it interacts with anything that has carboxyl groups at the terminal end and happens to be the right length. Well, look at malonate here. Carboxyl groups at each end, but this is only a 3-carbon strand. It can enter into the active site, and the carboxyl groups at either end can interact with the active site to an extent. But the active site of succinate dehydrogenase cannot react with this because it is not the correct length. It does not have the correct full shape. So this malonate can get in. It's considered an analog of the succinate. So it can get in, and if it's in and just kind of sort of interacting, succinate can get in. So we refer to this as competitive inhibitor. So as long as malonate is in there, succinate can't get in. Malonate gets kicked out, succinate goes in, dehydrogenase takes off. You'll get what is needed. Fumarate is going to give us malate. But here's the thing. The malate, I mean, look at this nice little dog here. We're going to get rid of this carbon-carbon double bond. We're going to replace those, those valence electrons with additional hydrogens. And, well, I'm sorry, 1 hydrogen, 1 hydroxyl. But we're not going to break water and add it in at once. No. What it is, we're going to add in a hydroxyl at first. So we're going to break this double bond. Carbon 3 here is going to get a hydroxyl. This is going to leave carbon right there. Carbon 2 with an open valence, a valence opening, which our hydrogen here is now going to enter into. So you end up with this carbanion. Remember, carbanion means you have a negative carbon with a negative charge sitting there. So here's our transition state. It's an intermediate. Fumarate, transition state, malate. What's interesting is, since you got that double bond there, well, cis-trans comes into play. Cis-trans, you either have fumarate or you have malate. Same thing once you have the malate. Which direction is the hydroxyl pointing? Pointing to the left. L, pointing to the right. D. It does make a difference. Because only L-malate can be turned into our final oxaloacetate. L-malate dehydrogenase, 1, 2, removes those 2 hydrogens. And you now have 1, 2, 3, 4 carbons, with carbon 2 having a carbonyl. But notice here, delta G 29.7. This is an extremely endergonic reaction. So a lot of energy needs to be poured in here. Even though this is considered a reversible reaction, the reaction is tilted left to right. Low oxaloacetate levels will push left to right. High levels become more reversible. But then again, remember, the oxaloacetate availability is what's going to push the citrate synthase. Remember, oxaloacetate has to enter into the active site first before the acetyl-CoA can then be activated on. So when you look at this, all right, energy, we're getting lots of energy. NADH, 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 so 3 NADHs, 1 FADH2, and then some substrate level phosphorylation for GTP, which will ultimately be turned into ATPs. So some textbooks, you'll actually just see it listed as ATP there. And then 2 carbons are released as carbon dioxide. What we are tracking, those 2 carbons that came in as acetyl-CoA, remember, circled those the first few times, they are now 2 of the carbons of oxaloacetate. 2 more will be added for citrate, goes around. 2 of the pre-existing that were in oxaloacetate will be released as carbon dioxide. And then on this 3rd trip around the cycle, that initial acetyl-CoA that we were tracking will finally be released as CO2. Somebody sat down and calculated this, and I don't quite get their math because I've tried to look it up and follow it, but all I do know is, yes, you have the substrate level phosphorylation and glycolysis and the citric acid cycle. You make a few ATPs. Great. The thing is, you're going to make 10 times more once you go after those excited electrons in NADH and FADH2. NADH drives about 2.5 ATP per electron. Because remember, you're bleeding off, and as you're bleeding off the energy, you're pumping hydrogens across them, from the matrix into that intermembrane space. The FADH2, this comes in one of the later cytochromes, about halfway through the electron transport chain, so that's why they're saying it's less. But the 2.5, the 1.5, I would never quote that. I would never hold you to that. Which is why when I look at these numbers here, I always cringe a bit because these numbers are going to be slightly different depending upon what source the author is reading, where they're taking their things from, how they're calculating things. So this is stuff I'm just not going to go and sit there and say, hey, you need to know this.